Hello everyone, welcome back to my channel By and for Byte. And in this lecture, we are going to cover the advanced Linux commands. There are different advanced Linux commands which we use to handle and visualize the biological data. So we will see how to use those commands, what are the syntax, and how we can use those things to fetch the data, biological data, and handle those kind of data thing. So what are those commands? So we are going to learn. So there are like commands like sed command, grep command, awk command, cut command, short, and tr command. These are very useful commands. And in this particular lecture, we are going to cover each command and we will see with an example like how to provide those commands, what are the options we can provide, and what are the outputs coming by giving those commands. And at the end of this lecture we will cover one exercise. So we will be handling two biological data. One is .gtf file, annotation file, and one is the genome file, .fna file. And we will see how we are going to use these commands to complete those tasks. Okay, so starting with the first, that is grep command. So what is the role of grep command? So you want to fetch any pattern from a file, you can use the grep command. And the syntax of grep command is you have to write the command name, that is grep. So there is an option. Options can be anything like -i, -e. There are a lot of options you can provide to fetch different tasks. Then you have to provide a pattern. So which pattern you are looking. And the next is your argument, means the file name. Okay. So the first example is like this is the command grep -i. The pattern I am fetching is Unix and giving the file name geekfile.txt. Okay. And within this file the content I have, so we already know to visualize the content of a file we use cat command. So this is the 4 line which is there in the geekfile.txt. Okay, now what I'm saying to my computer is I want to fetch a pattern that is Unix in this particular content. Okay, and given that option -i means I am making it insensitive. So for example, I'm not worried about the uppercase lowercase thing. If it is matching with unix, it will give me in the output thing. Okay, where when I run this particular command, you can see all the Unix word is highlighted. This lowercase also, if you have a small u and uppercase n, it is also highlighted because we make it with -i option. Okay, so this is the way to see like what are the all uh, contents containing this particular string, that is Unix thing. Okay, next you can also display the count, like how many number of matches you have in this particular file using the -c command. So -c is giving you the count for this particular pattern, and this is your file name. Okay, so the third important is like whenever you are using simply just grep, and for example you are not providing -i -c. Okay, you are just giving grep and your pattern and the file name. So what it will do, it will look for this particular string, and it can be anywhere. Like if it is a subset of any word, for example the first word is Unix arc something, so again it will show in the output because it is matching, the subset is matching with the pattern that you are looking for. But for example you are looking for a complete word, like you are not looking for a subset thing, you are grabbing the entire word, the whole word. So you have to use the option -w. So wherever this Unix as a whole word, it will come up in the output thing. Okay, now the fourth one feature for this grep thing is like you want to add a line. Okay, on which line this particular pattern is there. So as of now we had 4 line. If you see the gig file, so we have 4 line, and I want to see this Unix pattern is present in which line. Okay, so I'm giving grep -n and giving the pattern Unix, and because I'm not making it insensitive, so it will fetch for the exact match. Okay, so in line 1 and 4 we have the exact match of this particular pattern thing. So it was there in line 1 and line 4, so we also getting the line number. So using -n option you can print the line number for each pattern for each line. Okay, so these are the 4 features you can explore with the grep command. Now coming with the sed command. So what is the role of this set command? So you can use it to replace or substitute any string within a file. Okay. So same file I'm using it, and what I'm trying to say, I want to replace any particular string. So for example, I want to replace this Unix string and convert it into Linux thing. So how we can do that part? So we have to use the sed command, and the action, whatever action we are doing, we should keep it under code, either single quote or double quote, anything is okay, and need to provide the file name. Okay. So here I am saying I need to do substitute. So I am giving s and giving forward slash and providing first argument, which uh, uh, characters, or you can say string, I have to look on, and replace with which argument. So second argument is Linux. So I want to substitute Unix with Linux thing. Okay. So when you run this particular command, you can see this particular Unix is converted into Linux. So what is happening? Wherever it will find the first Unix thing, it will replace it with the Linux thing. Okay. So in each line, so first it find Unix at here, it replaces with the Linux thing. For the second line, there is no Unix thing, so no substitution happen. In the third line, again we have the Unix Linux thing. So the first matching thing is replaced with the Linux argument. Now is it jump into the fourth line, and it is looking for the Unix pattern, and this Unix is there. So again it replaced the first Unix to Linux. Okay. So this is the feature, like by default this sed command replaces the first occurrence in any pattern thing. Okay. So it won't replace the second and third or fourth or fifth. If you have the same pattern, it will not replace, it will only replace when it encounter with the first Unix argument, with the Linux argument. But how you can replace all, we have those option also in the set command. So first we will see how you can replace the nth occurrence. For example, in this particular file, you have 10 times this Unix character, and you want to
Ashish, Ashish, I want to ask something. Like I have a doubt. So, can you listen or just in this only? So, it was the first character Unix. So it was replaced. So if like U was in capital or some changes in the case of Unix was there,
hmm,
would that be replaced?
no, it will not pass. You have to keep the exact character.
Okay, 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 okay,
Now
got it.
replacing the nth occurrence. For example, in this particular file, you have 10 times Unix character, and you are looking to replace the sixth, uh, for example, sixth Unix character. So you can do that using this indexing thing. So for example, if you are using 1 2, so these are we call as a flag. So you want to replace the first flag, means first occurrence. You want to replace the second occurrence, you can use the second flag. For example, you want to replace the nth occurrence, you can use the nth flag. Okay. So same way, for example, in this particular thing, we are trying to replace the second occurrence. So we are using the second flag. Same thing, substitute which character or word you want to substitute with whom. So I am trying to substitute Unix character with Linux character, and which occurrence thing. So I am saying 2. So first Unix is reserved, so there is no change. When it encounters the second occurrence, which is changed to Linux thing, and the rest of the things remains the same. Now the third thing is, how can you replace the all of the occurrence of the pattern? So you can use the global feature. So G is referred as a global. So initially we were providing the indexing thing, or you can say flag, like you are using 1 2 3 4 like that. Okay. But you want to substitute in the entire place. So you can use global character, that is G. So the format is the same. You need to provide the sed command, substitute first argument, second argument, and what you want to do. You want to substitute one flag, two flag, three flag, or you want to substitute all. So the word you can use, the character is G for global replacement. So you can see everywhere the Unix was there, so that is replaced with Linux thing. Okay. Now there is one more feature is like, for example, I was saying in that particular file there was 10 times Unix is occurring, and you want to keep the starting three Unix character, but after that you want to replace it with Linux. So how you can do that part? So you can't do it manually like you can do for 4 flag, 5 flag, 6 flag, 7, 8. So you have to run the command for 6 or 7 times. Okay. So you can do it in a single go. What you have to do, you have to use this thing. So for example, from which flag you want to go global thing. So for example, from third flag, I want to substitute all the Unix with Linux thing. So starting first and two remain unchanged, and whatever occurring from third to the last one, that is replaced with Linux thing. So this is the important, if you want, like if you have an idea like I want to keep the first two information and want to substitute all the other information after second flag, so you can do it with this command. So there is one more thing, how can you replace a string on a specific line? So for example, uh, as of now in our file, this is a small file, so you can easily see like, okay, so there are 4 lines. So I want to replace, okay, in this particular line. So how you can do that part? So you need to provide the line name in the starting of the code. So simply, like for example, here I want to replace the Unix with Linux in the third line. So what I can do, I just need to provide the number on which line I want to substitute, and the remaining will be the same, and providing the file name. For example, you want to substitute in the fourth line, you just have to provide the line number 4. Okay, and it will replace your first character thing, your first Unix thing. Okay, now there are many features to delete lines from a particular file. So for example, this is the first syntax. So for example, you want to delete a particular line. For example, you want to delete the third line, so you can simply say D is basically represents here as a deletion thing. So within the code, I want to delete the fifth line. So you can say simply fifth. If you want to delete the first line, you can say 1D. You want to delete the second line, you can provide 2D. Okay. And if you want to delete the last line, for example, you have hundreds of line and you want to delete the last line. So you simply provide dollar symbol. It means the last line of your the file, and you want to delete because D is given, which means deletion thing. Okay. The third is if you are going to delete using a range. For example, again I was saying like you want to delete third, fourth, and fifth line. Okay, so you can't do it one by one. For a single line you can do, okay, I want to delete third line, I can use 3D. Okay, but if you have to delete third line, fourth line, and fifth line, how you can do? So you can use the range thing. So here in this case, I'm giving a range X and Y. So for example, I want to delete third to sixth line from my given file. So I can provide with a range X and Y. So this is like third comma 6. So from third to sixth line, it is going to delete from this file. Okay. And same way you can do like from nth to last line. For example, you already know like this is important to create a subset of file. So for example, if you are dealing with a file that is for example 2k or 3k lines, but to just for practice, if you are practice with the sum of the command line, it will take much time to process the and give you the result. So you have to subset the data. Okay, so you can practice the command. So how you can do that? You can simply retain the starting 5 lines to practice your code, and you can delete the other line. So same way, for example, set command you can use. I want to keep the starting 5 line and want to delete all the lines that is starting from sixth. I can give sixth, and this dollar symbol means till the last line, and I'm providing the deletion thing and providing the file name. Okay, so this will do the deletion thing. And how you can subset? You can use the append operator to convert all the output you are getting into a new file. So then you will have a subset of that particular file. There you will only have a 5 line of information. So this is useful to create a subset also. Like if you want to subset any big file, you can do that for like for internal purpose. Like if you want to practice something. Now coming to the last one, how you can delete a pattern. So same thing, if you want to delete a pattern, so we were discussing how you can delete the entire line from a file, but if you want to delete a pattern, so you can use same thing. You need to use the set command, and within the code you have to provide the pattern, and at the end you need to provide the D, that is for deletion. So here I'm trying to delete the ABC pattern. Wherever ABC was there in the file name, it will delete. Okay. Now the third tool is awk command. So where we can apply this awk command. So awk command has very important role. So you can read and scan file line by line. You can also split input files into field information. For example, if you are dealing with CSV file, you can split each input line into field of information, and you can also compare input fields with any pattern you are looking for, and you can perform actions on the match line. For example, if you have a specific pattern in a particular line, you can filter out those particular line and you can perform many actions on that particular thing. Okay. So the syntax for the awk command is simple. So you need to provide the awk command. There will be a lot of options. So option will be there for each command. You have to know how to use that, which option is used for which purpose. Then there is an within the code you have the pattern selection criteria, means you are looking for pattern, and within the curly braces what action you are going to do, and then the argument means input file, and this append operator is like whatever you are doing here you can simply print on the screen, or if you want to store those results into a new file you can convert into output file. So you just have to provide the append operator. So I was saying same thing like previously, if you want to subset a file, you are doing every action on the left hand side, you are giving append operator and you are storing all the results in this particular output file. Same thing, you are running the awk command, doing some selection and doing performing some action to do some filtering, and then you are storing those results in an output file. Okay, so let's see with an example. So this is the default behavior of your awk command. When you are going to just say awk and within the bracket, I'm not providing any selection pattern. So I'm not looking for any pattern. I'm just saying within the curly braces, the action item. Action item is print, means I want to print this entire content of this file. So this is the same thing what we generally do with the cat command. Okay. If you do cat and then provide the file name, it will print the content of the entire file. Same thing awk is doing if you're not providing any selection thing. So if you see the syntax thing, we have the awk command options and selection criteria. But here we are not providing any selection criteria. We are just giving the action item, that is print. So it will print the entire content. Okay. Now we want to fetch with a pattern. Okay. So for example, here I'm saying awk, and within the code, this is the selection criteria or pattern, and this is the action item. So I'm saying I want to fetch for this manager pattern and I want to print. So I want to print the output on the terminal, and this is my file from where we are looking. So if you see, we have these many rows and only the first, third and fourth has the manager information. So in the output you will get these lines. So this is like how you can filter out any particular line having these particular information. So you are going to say where I can use that part. So for example, when you are dealing with any annotation file, so annotation file will have a lot of information, like it will have information for the regions for exon, intron, coding region, non-coding region, and for example you are only interested for coding region, so you can fetch that coding region pattern and then you can subset those files. Because if you are going to work only on the coding regions, you can provide the selection thing, I'm only interested for coding regions, so the pattern will be the coding region you want to print, and provide the file name. It will give you the output of all the lines which has coding region. Okay, now how you can split a line into fields. Okay, so in awk commands, if you want to print any particular column or you can say field, so you just need to provide the indexing thing with a dollar symbol. Okay, so here I am saying print $1 $4. So I am saying the first column and the fourth column. Okay, so here you can see we have the fourth one, two, three, fourth column, and by default, if you're not providing any delimiter, like whether the file is TSV, CSV, what separation is there, so it will consider space as a separator. Okay, so because we have a space in this txt file, so it will consider as a four field. So first field information will be the name, second this is manager, third is the account information, and fourth is this integer value. Okay, so we are saying to print the first column and the fourth column. So you are getting in the output the first column information and also the integer column information, because that is the fourth column. Okay. Now the next thing is there are many inbuilt options in awk command. So there is first one, nr thing. Okay. Nr means you want to print or display the lines. So if you want to have also the line information, you can use nr command. And so here is an example. I'm giving print nr comma $0. So $0 means you are printing the entire line. If you are giving indexing 1 2 3, it means like you want to print the first field, second field, third field. But if you are giving $0, it means you are printing the entire line. Okay. So in this particular command, what I'm saying just print the line number. So it will print all the line number 1 2 3 4 5, and I'm saying print the entire line, so it is printing the entire line. Okay, for example, you want to print only the third to six, so you can provide like you can provide the line number, like nr is basically for the line number, displaying the line number. So if it is matching with 3 and 6, from 3 to 6 I want to print the line number and the entire line content. So this way you can subset the line also. Next is enough built-in variables in the awk command that is used to display the last field. So whatever field information you have in your file. So for example, if you have 10 fields in your file and you want to print the last field, you can use simply the nf field. So you don't have to open the file and see, okay, this is the first index, second index, third, fourth, fifth, sixth. If you are interested in the last index, you can simply use nf thing. Don't have to provide, like for this example, last index is 4, so I have to write $4. So instead of $4, the alternative option is you can use nf thing. So here is like awk print $1 means first field information, and $nf means the last field information. Okay, now the next command is cut command. So as of now we have learned grep command, awk command. So these are very similar, like some minor difference, and you have to learn how to use that. When you do practice, you will remember those commands and you will know, okay, for this particular purpose I can use grep thing, for this particular purpose I can use awk command. Okay, so this will more clear in the last slides when we are dealing with an exercise thing. Okay, so cut command. So cut command in Linux, how you can use it. So the syntax of cut command is simply the cut command and providing the options and the file name. Okay, so this is an example where we have a file state.txt, and this is the name of the file. Okay, Andhra Pradesh, Arunachal Pradesh, and these are the name of the files. Okay, if you just simply do cut and provide the file name, it will give you an error. It will ask you have to provide the options also, which is mandatory. So here the error, you must specify options, either a list of bytes, character, and other field information. Okay, so minus b is used for the bytes, minus c is used for the characters, and minus f is used for the field option. Okay, so in this particular example, I am saying I want to print the starting three characters. Okay, so cut minus b 1 comma 2 comma 3. So it will print the starting three characters of each line. So for a and d is getting printed for the first line, aru is getting printed for the second line. Respective same thing. Okay. Now if you see for the, if you want with a range, like I want to print the characters from 1 to 3 and 5 to 7. I'm skipping the character 4. So you can see Andhra Pradesh, you can see the first A and D is getting printed, H is replaced, and H is not there in the output because we are not considering the fourth character, and we are printing fifth to seventh. So we are getting 5, that is R, 6, that is A, and 7, that is space. Space is also considered as a character. So that's why we have this information. Same way, this is printing for the other row information. Okay. The next is I want to print from the beginning to the end of line. You can simply use one dash. So wherever you see dash means it is going to the end part. Okay. So I'm saying from the first character to the end of the character. I want to print. So it will print the entire name. Okay. For example, if you're going to say I want to print from -3. So it will say from starting till 3. So like from starting, and I want to print till 3 characters, you can use it. If you want to print till 7, you can use -7. If you want to only print the for example, this first row, the state name is Andhra Pradesh, so you want to get the output only the Pradesh part. So what you have to do, you have to count. Okay, this is like having how many characters? 6 characters, and there is one space, 7 characters. So I want to print from 8 and give dash. In the output you will get this particular thing, and this will not be there in the output thing. Okay, so this is the way you can print a specific line characters thing. Now -f is very useful. I generally use this particular command. uh, if you have watched my first video also, though I have started with a question, that CSV file, how you can fetch any particular position. So there I have used the cut option, cut -f. Okay, so what I was doing that time, I was giving the cut command. The syntax is cut command, there is an option. So whatever, so -d is one option, -f is one option, and giving the file name. Okay, if you are going to provide the delimiter, it will consider, for example, if you are dealing with CSV file, you need to provide the semicolon. If you are dealing with the TSV file, you need to provide tab delimiter and which field number you are looking for and the file name. Okay, so here for example, in this particular example, I haven't provided the delimiter thing. Okay, so I'm just saying cut command and -f providing the file number. So I am saying cut -f1 and state.txt. Okay, so it will consider everything, it will print everything because we are not providing the delimited options. So it will consider space as a separator thing. Okay, so it will print everything. If you are providing the space as a delimiter, as a -d option, then only it will consider, okay, space is there, so it will print the first information, the first field information. If you are not going to provide the -d option, it will print the entire content. It will not consider like there is any delimiter is there or any separation of the words, like whether you are dealing with semicolon, tab separator, or any space. So you need to provide while fetching those information. Now this is the I guess second last tr command. So how you can use the tr command? So tr command is simply used to convert lowercase to uppercase and also like reducing the space thing, everything. So the thing, so tr is like truncated part, something you can remember. So tr, and you have to provide the option, and there's two set1, set2. Okay, so for example, this is a file and the content within this file is this one. Welcome to is written in the uppercase and the second line is written in the lowercase. Okay, so I want to convert everything in the uppercase. Okay, so what you can do, you can do simply cat Greek file. So whenever you are going to combine two commands together, you have to use a pipe. So this is a pipe option. So what you are trying to say, so you are saying first display the content of this file and then implement this particular second command. So what it is doing, it is like uh, this truncation is happening within this particular thing. So welcome to is there, and we are saying the set1, all the lowercase from a to z should replace with all the uppercase from a to z. Okay, so this second line having the lowercase is replaced with the uppercase, and then we are printing those particular file content. Okay, so we are combining two commands together with a pipe command. Okay, there is a second option also. We can do that part is using within the bracket you can provide the colon and giving the lower and again colon, and then you are saying all the, if there is any lowercase in particular file, it will replace with the uppercase. So both of the command will do the same thing. Okay, so where you can use this particular thing, for example, sometimes while fetching the gene information, sometimes your gene name is written in lowercase or uppercase, and so you make it constant. Either you can replace it with lowercase or uppercase, so you can, it will be easy to annotate with your when you're dealing with annotation part, like how you are going to annotate your genes information with the reference genome, so you will get the count of each data for each gene. So that time these commands will be useful. Now how to translate white space characters to tabs. So this is simply you can use the same thing. The first set is like I'm saying all the space should be replaced with the tab character. Okay, so here you can see there is a space after welcome and two part, so this is replaced with the tab thing. So you can do that part also with the tr command, and this is very important. So if you want to replace anything from a file, you need to provide both the argument. So for example, in this particular file I am replacing the curly braces with this close bracket thing. So you just simply have to use tr and provide both the argument and provide the file name. If you want to store it into a new file, give the this append operator thing, and you want to store in a new file, new file.txt. So what will happen? So this curly braces will replace with this close bracket. So we generally use this particular thing. I prefer to use this particular command when I'm dealing with, for example, if you have a lot of information. If you ever encounter with VCF file, so you will see a lot of information like gene information, type, then this is a hotspot variant or non-hotspot variant, then the score. So all the information will be there in one row, and all the value will be separated by colon. So it will be difficult to see the value of all the information. So what I generally do, so I separate the semicolon with new line. So after applying those condition, I will be have all the information in a new line. Like for first information will give me the type of gene, second information or second row will give information on the hotspot non-hotspot variant, third will give v score thing. So it will be easy to visualize the content of a file thing. How to delete any specified characters using -d option. Okay, so for example, again I am combining two commands. So echo command is going to print this particular thing, and within this particular thing I am saying truncated command to replace -d with w. So sorry, I'm going to delete so this is -d's option, and I'm going to delete the first wherever w is there, it will replace, delete the particular thing. So here you can see we had this welcome to Geeks for Geeks, but we are deleting with tr command, and we don't have w in the outcome thing. Okay, now coming, and let's see with an exercise like how where you can implement those commands and how you can use that. So that will be very useful. So let's see the problem. Okay, so how you can deal with any biological data, and whatever you have learned, how you can implement those commands. So I hope like even I told you like you can keep those two files. So if you are dealing with genome build, so you all the information related to chromosome will be there, you can download from ncbi, and the extension of the file format is .fna. Okay, and if you are dealing with annotation file, so either you can have the gff file format or gtf file format. Both are considered as an annotation file. There are minor difference in the formatting thing, but the information will remain the same. Okay, So today I'm going to deal with these two files. So these are the biological data and how you can do all the steps. So what are the steps I'm going to do? So our target and goal is like download the GTF file. When you download the GTF file, it will be there in the .gz format. So you have to unzip the file. Then what I want to do, I want to fetch all the protein coding regions or genes, okay, using a grep command. Now I want to convert all the semicolons which are there in each row with a new lines. Then the next target is I want to select all the gene name, and I want to see how many genes are there that have this protein coding regions, okay. And then I can sort, okay. Sorting means like we can sort in alphabetical order, and then we can find how many unique genes we have in that particular annotation file, and then we can count those things. So starting with the first problem, so how this genome file looks like. So if I want to see the starting content of any file, I have to use head command, head minus n. I want to get the first 15 line, okay. So this is my fnf file. So initially I have downloaded, it was .gz, but because it was a big file, so I unzipped it before the class. So this is my file. If you print enter, see, you have all the information. So because as of now I am printing only the starting 15 lines, so the first line is giving information. So all of the fnf file is starting with greater than symbol. Then we will have identifier, means like this sequence is coming from which sample. So this is the sample of Homo sapiens, and this particular sequence is for chromosome 1, and these are the other information, okay. Now the task is how you are going to add another genome in this particular file. So this file contains human genome, so chromosome 1 till chromosome 23 and other mitochondrial content, okay. But for example, if you have done any wet lab experiment like plasmid thing, like you are inserting external genome into your plasmid thing, for example, so you will have also external genome. For example, you are inserting any bacterial genome, then you will also have a bacterial genome in the entire build. So how you can create that part? So we are going to solve things. So first thing, if you visualize the entire file, it will take a lot of time to open. And the second thing, how you are going to validate all the chromosomes are, or all the sequence, reference sequence for the chromosomes are there. So we will verify that part also, and then we will create a new file that we can say a bacterial genome, and that we are going to combine this bacterial genome with this human chromosome genome thing, and then we will have a build which consists of human genome information and then bacterial genome information. So now the file is loaded. So for example, this is for chromosome, this is an NCBI ID for chromosome 1, and I want to see what is the name for the chromosome 2. So you are scrolling down. It will be difficult to find the information, or you can say the identifier information for chromosome 2. So how you can achieve that, okay. So I'm just closing this file, and just we have learned the grep command. Grep command is used to fetch the pattern. So what I'm trying to say, we know the identifier start with greater than symbol, okay. So I'm fetching that particular pattern and giving NC, okay. Within the bracket I am fetching the pattern, then providing the file name. The file name is this one, .fna. It will print all the rows which are starting with greater than symbol and ANSI character. So you can see ANSI 1, this is for information for chromosome 1, chromosome 2, 3, 4, 5. So okay, we are knowing that okay, all the chromosome information sequence are there in this particular file. By mistake, if you have downloaded any other file that is lacking with chromosome 8 and you are working on chromosome 8, because the data you are working is going to map with chromosome 8, then this will create an issue. So you have to verify whether your file that you're going to use have all the reference sequence for all the chromosomes. So here I have printed. So the file contains information for chromosome 1 till 22 XY and some information of mitochondria. Okay. The file also contains scaffold information, but because we were fetching with ANSI and the scaffold information is with different things. So how you can fetch that part also? You just do an or greater than symbol and fetch all the information or all the rows which are starting with greater than symbol. Now you can see there is anti also. Anti is reason for the scaffold. So the naming convention for the scaffold regions is anti. Okay, just give me 1 second. Yeah, it is still running, and then you can see all the rows which are starting with greater than symbol. So you can find, okay, the entire genome contains which information. So it has the scaffold information, it has mitochondria information and other information, and also the chromosome information. So this is the first task we have covered, like we have find out, okay, so this file contains all the chromosomes that we needed. Now the next task is I want to combine. So how you can do? So you can create a new file, touch, and for example you can create bacteria.fna. So this particular file will contain the bacterial genome. Okay, now I can open this file simply, and as we know the format for any fnf file will start with greater than symbol. So I need to insert I greater than symbol. I can say this is bacteria genome, and then I can write any sequence A C E. So for example this is the sequence I write. For example I am writing, so this genome is like this much sequence this has. Okay, I'm just saving this particular file. Now how I can combine both files together? I can use the cat command. You can provide the first file name. So what was the first file name? So it was your gr.fna. Second file that you have created is bacterial genome, and you are going to combine both file in a new, that is mixed genome new.fna. So what it will do, it will combine all the information in both the files. So in the output file, you will the output file will be the mixgenome underscore new.fna, and this file will contain information for the human genome and plus bacterial genome. Okay, so it will take some time. So we'll wait 1 or 2 minutes. It will complete, and then we can see the content of this particular file. So any doubt for this exercise we are doing, like as of now we have covered how we can see the FNF file and then how we can create any other genome file. Now we have, you can see we have mixed genome new FNF file. Okay, if you again do the same thing that we were doing to see what chromosome information we have, we can use the grep pattern / and providing this mixed genome right now. Okay, so it will print the entire uh, header information for each sequence that we have in this particular build. So we just have to verify whether we are getting bacterial genome at the end or not, because we have added. So this is the thing we have added. So now we have the bacterial genome also within this particular file. So now in the mixed genome we have the human chromosome information and also the bacterial genome information. Okay, now the next thing is now we can see with the annotation file. So for now, so this GTF file, So this is a huge file. So what I did, so I have created a subset file for testing. Okay, so if you see, so I have this particular file. So this has a lot of information. But if you process each command one by one, it will take a lot of time to get the output thing. So for this demo classes, like I have created and subset this particular file and only keep it like 30 to 40 rows. Okay. So same thing, we have this testing.gtf file, a small annotation file. Okay. Now what we are going to do, we are going to solve the exercise one by one. Okay. So what was the first exercise? Unzip the file. So we already have the file, and now we need to select the protein coding genes with the grep command. Okay. So we first see. Let me open some starting line to see the content of this particular file. So testing.gtf. Oh, sorry, head. Oh, I am giving it. Yeah. So to make it simple, give it 5. Yeah, we can. So first 5 information is hashtag another information. Yeah. So these are generally information in each GTF file, like the description thing, like the source is GenCode and where the format is GTF thing, when it was uploaded, everything, and what is the version and human genome. Okay. And from the fifth line, we have the information like chromosome, then gene name, starting and position of this particular gene region, and other information in symbol ID, gene type. Okay. Now we are looking for this information. Whichever rows having this protein coding information, I want to extract those information. Okay. So that was the question. Select protein coding genes with grep. So because in this particular file we have a lot of genes, but I am only interested in my project. The task is to deal with the protein coding regions or protein coding genes. Okay. So I have to extract those genes that is protein coding. Okay. So what I will do, I can simply do grep and protein coding. So and giving the file name testing.gtf. So this will show. So initially we have, for example, 30 and 40 is rows, but now for example we are getting 10 and 12 rows which are matching with, because we have the information for protein coding. Okay. So what we have to do, we can create a subset of this particular file. How we are going to do that? So for example, this particular thing I want to store in a new file. So just name it testing new one .gtf. Okay. Now do ls. Now if you see cat testing new one.gtf, you will have all the information and all the genes which are only having the protein coding part. Okay. So we have created a subset file with the genes having protein coding regions, or those genes are protein coding. Okay. So the first task is done. So we have the file and we have the genes with protein coding thing. Okay. Now the next thing is convert semicolon to new line. Okay. So what exactly the meaning is? So you can see all the information are there in one row. Okay. So we have a many field. First field is for chromosome information. Second field is the name. Third is the gene. Fourth is the start position. Fifth field is the end position. There are the information. And same way, this field, for example, 7 or 8th field has a contain multiple information like gene ID, gene type, gene name, level, and other information. But we are only interested for the gene name. So what we will do, we will convert this semicolon. You can see here this is semicolon. I want to convert this semicolon to new line. Okay. So how you are going to do that part? So to convert from one character to other character, we have learned the truncated. If you remember, like that time I was saying, parenthesis was converted into close bracket. So I'll use the truncated method or truncated command. So how I can do? For example, I want to show the output to you on the terminal. So I am saying testing. So this file was, which file this was? Testing new one GTF. Okay. So cat testing new one.gtf. Because I'm combining multiple command, I'm using pipe command. Okay, I'm using truncated command, and what I'm saying, wherever semicolon is there, I want to convert it to new line. New line is written in the Linux is /n. Okay, press enter. So do we have missed out anything? Cat, this one is there. This cat testing new GTF, and then we are doing truncation thing, new line. So let's. So let me store the results in a new file. So give it a. So I know this. Should not take much time. Let me do it. Okay. So I can visualize the testing file, and then we have the subset.gtf, and then we are combining with the pipe thing, and we are doing truncated thing, and then we are using colon space, then new line for this character, and press enter. Yeah, so maybe some typo I was doing. So you can see. So what I did, so I have this file. So I'm opening and visualizing the content of this file with a cat command, and then combining with a command that is truncated, and where I'm saying replace the all the semicolon with a new line. So you have seen, so after gene ID we have the semicolon, then after transcript ID information we have the semicolon. So what is happening? So this gene type we are getting printed, then this is going to a new line. We have the gene name, then there was semicolon. So we replace it with the new line. So the other information will jump into the new line. So we will have a, like, we can easily visualize the information for each row. Okay. So the second task we did, like we have converted the semicolons to new lines with the tr command. Now the next thing is we have to fetch the gene name. Okay. So how you can do that part? So first let me store this results into a new file. So I can do testing subset underscore one.gtf. Okay, so we have stored this part, and now we can grab this gene name. So now our target is to grab the gene name, and from which file we are looking for, testing subset.gtf, testing subset one dot gtf, and what we are trying to do, so first we will grep. So see, so we have these many genes, so all the gene information are same, okay, like because I was like I have taken only a small subset, so all the information was related to this particular gene, okay. So for example, let me do one correction, so I can open this particular file for now, and I'm going to replace some of the gene name with a different name, so it will be easy to see the variation thing. So I replace it with cr, and for the next I did p, and for example again O, I make it start with T. So now we have a 4 different genes information in this particular file. Okay. Now again you fetch the grep thing. So you can see like you have these many gene which you have sorted based on the protein coding regions. So now you will see, okay, so you are trying to see how many genes and which are the gene types that are there in the particular file that you are looking for. That is basically the protein coding genes. Okay. So now we have the list of protein coding genes. Okay, that is CR4, 5, then PR and TR, then OR thing. Okay. Now the next task is we have completed the third task. Now I need to sort it with alphabetical, because when you're doing with a big file, you will get a lot of genes like 5,000, 6,000. So you can arrange in an alphabetical order. Okay, so how you can do that part? So in the same command, either you need to store the output of this command into a new file, or if you want to do it in the same thing, so you just have to again use pipe command, because you can combine many commands together. So you just have to use the pipe command. So what I'm trying to do, I'm grabbing this gene name first thing, then I'm saying I want to do sorting. Okay, when I press enter, the gene name is sorted. So starting with C alphabetical, this comes first, then all the genes having O name is coming second, then P, then T. So the list is sorted. Now the next task was how to find the unique gene. Okay, so I want to know that in my database how many genes are unique. So what you can do that thing, so you can simply within the same command, you can do unique is command and using -u for the unique part and press enter. So you can see these are the three rows. Yeah,
Ashish, I have another class. Like,
yeah,
I'll
you have
just,
to do the recording.
yeah, yeah. I'll continue from the comments.
Yeah, yeah, sure.
Yeah, bye. See.
Yeah, so I was saying, as of now, we were trying to cover this things. So first thing, what we did, we have a big database. We have a big gene information means annotation file. So I have to filter out all the genes that are protein coding. So we did it. We did with the grep command. Then additional information we have in that particular field. So what I did, I converted all the semicolons to a new lines. Then from that particular file, I fetch the gene name. Okay. Now I have a list of gene name that are there in the database. I have sorted with the sort command. Now my target is to find the unique genes, means the occurring of those genes is only once at a time. Okay. If you see, like we have these gene names, so only these 3 genes which are unique. So when you add this unique command also in the output, you are getting this 3 thing. Okay, because we are doing the subset, so we are getting 3. If we are covering the whole exercise with a big file, that the complete gtf file, we'll get a lot of rows information. Now the next target is count the unique genes. So we can easily count like it is 3. But if you are having a like bigger number of output in the row, so how you can do that part? You can simply combine wc minus l. So there are 3 rows, means total 3 rows we are getting. So you can see, like if you are getting a lot of 1k genes after running these commands, so you can't count the entire rows, so you can simply use the command to count. So this is the task count unique genes name, so we have used the wc command. So like for example, for the last 3 and 4 command, I was not storing the output in a different file. So I have just shown like how you can combine all together using a piping method. You can also do, you can grab the things, store in a different file, then you can do the sorting thing, store in a different file, then you can do unique thing, store in a different file, and then you can count the number of rows from that particular file. Or if you are comfortable and good enough to use the piping thing and combining many commands, you can do that part also. So this is the whole exercise. So I was trying to say like there are many biological data to deal with, and to find your answer, you every time you can't open the file and see and finding things. To make it easier or to make your life easier, we have to depend on the linux command to make the work faster. Like simply, for example, I have to look for the protein coding genes. How you are going to do it here? You are going to search, okay, find, and then you are going to search protein coding. Okay, so this is the first row. Okay, so you're going to see, okay, this is the protein coding. You're going to find next, okay, this is the protein coding, this is the protein coding. So this is a difficult task and tedious task. If you have a small number of subsets, so you can do this thing. If you have a lot of information, for example, the entire genome file, so you can't imagine like how many rows it has. So to deal with, you have to rely on the linux command, and these are the very good and advanced linux command, but you have to practice how you can combine those commands, when you can use the grep command, when you can use the cut command, when you can use the awk command, when I have used the truncated command, when I have used the sorted command, what is the role of unique command. You have to practice, and you can practice with other biological data, like try to practice with vcf file. Vcf files has other information, try to fetch those information. Are you able to fetch those information or not?
So
Try to
please like and
work
subscribe
with
my channel
FASTA
and
file,
also press
try
the bell
to
icon
work
to
with
get the
FASTQ
notification for
file,
the further
try to
lecture.
work with BAMSAM file, and see if you are able to fetch the information that you are looking for.